where the phytoplankton is.
Thank you all for these wonderful presentations. This question is particularly for Adam and Aaron. There's some weak and laboratory-based evidence that grazers select for nitrogen and phosphorus-rich prey. I was just about it's wrong. Okay. So there's some laboratory-based evidence that grazers select, or phytoplankton select for nitrogen and phosphorus-rich prey, and that would sort of put the results that you've shown, put those kinds of phytoplankton at risk. What are your thoughts on that?
All right, so it's true that, okay, so it's true that there's some early experiments like Bob Sterner and others sort of promoted this idea that grazers don't like high C2P, like prey with high C2P. I think it was Daphne, if I remember right, a lot of the work has been done on. And I think that, first of all, what I think we're really missing is that there's like a tremendous diversity of grazers. And I'm sure that there are some grazers that like prey with one chemical composition, and there are other grazers that like it with different elemental compositions. It's not like at bats where we have sort of a rather extreme chemical composition of phytoplankton that there's no grazers around. And second of all, I think that you can't do just a direct translation from the predator to the prey, because the predator will lose a tremendous amount of carbon to respiration, and different predators will lose much more because they're moving around, so they have a big respiratory cost. And so I think the idea in ecological stoichiometry of linking predator and prey elemental composition, I think we need a lot more knowledge and data before we can go to that. And I think the question of how grazers influence the chemical composition of exported material, I think is a wonderful research question that we should do a lot more on. And I think the diversity of grazers, how they play a role in that, I think is also an amazing question. So I don't think that we have such a great evidence that food quality is related to the elemental composition yet. But I rather think that there's just a lot more going on in the ocean.
Yeah, I'll just add that I think there's some really interesting preliminary evidence that iron content in these cells can also potentially be a driver. But yeah, many more pieces of data are required, I think, before we can understand what that means for balance between growth rates and consumption rates. But I think it's a key question that we should be paying more attention to. Thanks.
I had a question that sprung off of Richard's concluding remarks, and I'm hoping, Richard, you can elaborate on this as well as others, and that's that these storage pools of nutrients that you're observing, you explain that they allow organisms to exploit more variation in resource availability, and I just was hoping you could elaborate on that a little bit.
Yeah, I did. I had a little slide that I was going to show about that. I have a feeling now that a lot of the core metabolism is highly conserved, and that organisms respond to the limiting factors. And there was a question that was raised about alkaline phosphatase, for example. So under phosphorus limitation, they'll do that. But the other thing is, I think there's more of a role for these stores, for iron stores or phosphorus stores or nitrogen stores, as a way of buffering against environmental change and as a way of taking advantage of any episodic availability of resources. And it kind of grows out of my interest in light. And when cells are clearly moving through a light gradient, and when they're up near the surface, they can grab the light and store it as an energy reserve, and then they can mobilize it when they're down at night or in a darker area. And they use that energy reserve for something, and they use it for accumulating nutrients and accumulating biomass. I think different organisms are going to have different potentials. So it depends on how they can store the nutrients. And I guess to the extent that they are actually nutrient limited in their environment. I don't know if that goes far enough. But
I was thinking too in environments where there might be sort of really truly chronically limited for a nutrient, in low iron systems or low nitrogen, some of these oligotrophic gyres versus environments that are seeing more of these pulses coming in.
I think that variability in the environment then becomes a selective pressure. And it's a question of whether you specialize in acquiring the scarce nutrients or taking advantage of the pulses. Related to what Stephanie was talking about in terms of size and the way in which organisms obtain nutrients in the gyres. There are lots of ways in which phytoplankton can obtain nutrients in the gyres, and you don't just have to be small and efficient at low concentrations. There's vertical migration, there's mixotrophy. There are a lot of strategies for obtaining nutrients there. In higher nutrient regions, it's going to be the ability to sequester nutrients to prevent other organisms from getting them may give you an advantage. So there's a lot that we can play with there in terms of the physiology and then the interaction with the ecology.
This is a question largely for Stephanie. So yesterday, the gauntlet was thrown down that it's all physics. And we know we have really large biases in nutrient delivery via physics in our Earth system models. You've highlighted these adding functionality as really changing things regionally, like locally. But can you talk about plankton diversity and functional diversity relative to the biases in physics? How important are these things for larger biogeochemical cycles like carbon export, for example?
Well, I don't want to steal Ben's thunder, but he's shown that mixotrophy can significantly increase the global export production. So it's better ways to utilize the nutrients. Keeping them at the surface by eating something before it can sink. So I think there is a biological control there that is modulating that physics controls everything. I think there's a piece on top of that is the biology finding unique ways to be able to trap that nutrients. For instance, things like buoyancy control could be another one that could be added to try and keep, get those nutrients back up to the surface. So I think physics sets everything up.
Yeah.
Biology then tries to mooch. It figures other ways around it to try and compensate for the physics.
So quick follow-up. So then is it more that functional diversity that's important, or do we need like a bunch of different diatoms, for example?
So DDAs are finding a way to get nitrogen. So you don't just need the diatom. I mean, you don't just need the diatom, you need the dinoflagellate. If larger cells are responsible for much larger amounts of export, then you need the size as well. So I don't think I can answer your question. I think both.
To be determined, perhaps.
Hello, yeah. This is a question to all of you. If, from a modeling standpoint, what are the 3 most important experiments we should conduct?
I'm going to not answer that because I want to conduct those experiments too.
Well, one of them is the multifactorial thing that Bill was already making everyone nervous about. How do we put those multiple limiting factors, and not just nutrients, but light, temperature, together? It seems an incredibly daunting task, especially when I hear that just 2 of them is 3 years of somebody's life. But it's those planes that I think we need to know. It's those interactions, and more to the point, whether they're universal, whether we can find from just one phytoplankton what that plane is, as opposed to having to know for every single species what that is. So that would be one. Does somebody else want to take 2?
That's a daunting question, I gotta say. Well, one of the questions in phytoplankton experiments that we struggle a lot with is, at one hand, we want to replicate that there's a whole community, right? It's not just an individual organism, which is what most people are looking at. They take one organism, they manipulate a whole range, and then they look at the outcome. But of course, there's like all kinds of interactions. The problem is, if we have a whole community that you put in and you do manipulations, you see a big community shift. So how do you do a good experiment where you don't see any community shift, but on the other hand, you manage to manipulate an environmental gradient at the same time? I think that's a really relevant question, but also a real challenge for how to do sort of physiology, if you want to call it that. So maybe it's not exactly addressing the question, but just answering another one, asking another one. But nevertheless, I think, and also a lot of manipulations are rather extreme. And so therefore you do see big turnovers in the community. So how do you sort of do these sort of subtle changes without sort of like getting real crashes in the community? Those are experiments I would like to see.
Yeah, I guess there are loads of different experiments that you can do, but it depends on the context in which you want to do them. Do you want to do an experiment with a natural assemblage, in which case you want to minimize the perturbation, but you still want to see a response? You want to do something realistic there. The kind of bottle bioassay experiments tell us a lot, but they are also very difficult to interpret because we don't know just where the limiting factor is kicking in. I think in the laboratory, what we just want to do is identify fundamental processes and get some idea of the responses. But then because of the huge diversity of organisms, the allometric effects, the other effects, I think we can only use them as a little bit of guidance for the way to think about things. They're not going to provide definitive answers. They're going to provide insights.
Just add quickly to that one of the ways in which I sort of select experiments to focus on in the lab is by looking at the predictions from the geochemical measurements. So why focus on iron and manganese in the southern oceans? Because the ratio of iron and manganese to other macronutrients are suggesting that there should be interactions there. So that's one way to focus on what are sort of underexplored interactions that might be important. So looking back to the geochemistry for suggestions on your biology, that's sort of how I've been approaching it.
So my question is in relation, it's a bit of a follow-up from what Richard just said in relation to these bioassay experiments, especially in the field. I love them, I do them all the time, but as I'm sort of, or as the community is working or moving towards more molecular indicators, what we've been finding is that we're seeing essentially different responses at some points when we use molecular indicators versus what we see when we're doing more of the traditional experiments, because those manipulation experiments require an increase in phytoplankton biomass in order to interpret what's going on. But usually that may not have been the limiting nutrient in situ, which we can assess from the molecular indicators where we're not doing that same perturbation. And I think that's where the initial biomass, the T0, becomes very important in interpreting what happens after a certain period of time in incubation. So in that regard, as we move towards looking at multiple stressors and doing these factorial design experiments, how do we better interpret these responses, especially given that you could end up with a situation where one factor wasn't limiting initially, but just became limiting because of the increase in phytoplankton biomass as you perturb the system. So if somebody wants to comment on that. Yeah.
I think I can start. Adrian, that's something that I stress myself out about a lot. And one of the reasons why I started doing transcriptomic sequencing on those bottle incubation bioassays along a time course, in order to convince myself that, or in order to understand what that progression from the initial community through inducing a limitation, what that response looked like at the molecular level. And now I think we have an accumulating body of information about that response over time, at least at the transcriptome level, to begin to feel more comfortable interpreting those biomarkers in the context of the field. So the only reason I felt good about showing you that data that I showed you today is because I spent all that time staring at a time course transcriptome when we added iron, when we added B12. So I didn't want to bore everyone with those details, but I think it's extremely important to pay attention to and to remember that we're looking at really different scales and really different biochemical questions when we're comparing a bottle incubation bioassay to this. So yeah, it's an excellent point. I think those time courses are really important. And we were getting to the point where we can do the same thing with proteomics now on smaller volumes, so we don't have to use giant cubitaners anymore. So I think there's hope for more data to understand that time course concept a bit better and help us interpret more. But it's a really important point.
A question here. So across the talks we've heard in this session, we've seen the very potent and powerful combinations of molecular biology or ecological stoichiometry with physiology. And so in terms of a longer game to try and integrate those into modeling, would you maybe like to comment on some long game approaches we might take? So for example, metabolic flux modeling or dynamic energy budgets that other groups have had to play around with. There's still progress to be made in those fields, but I'm just wondering if we're looking at, say, over the next decade, is there some way we can integrate the requisite detail you guys are showing from physiology, omics, and elemental stoichiometry?
There has been, I think there's an ongoing need for some of these things like this macromolecular approach that Richard has worked on, but moving forward in that and using those levels of models to help us parameterize processes within the model, so whether it's a question that we can carry, within the cell level, can we begin to understand the pathways that set up stoichiometries, that set up the final growth rates, and then can we distill that into something that's tractable to put on our models? So I think the macromolecular approach is probably a very an important one. Whether the flux, the FDA type models, those become incredibly complicated. Again, I think it's a matter of extracting out from them the key points and being able to parameterize them. So I think all of these steps of increasing complexity are important, but getting them into models requires distilling the information and parameterizing.
And so, for example, just to follow up briefly, would you see the value of sort of some offline runs in the way that we made a lot of progress, for example, going back to some of the synthesis and modeling that came out of JGOFs, some of the online runs that people like Keith Moore and Scott Doney did really helped to bring multiple elements and some more detail into models without the issues of computational expense?
Absolutely, and I suspect there are other people out there, but I'm just thinking John Casey at the University of Hawaii has a flux balance model looking at Prochlorococcus, and then he's been trying to start running that in using model output of temperatures, nutrients, etc., and seeing how that the FBA progresses into the future. So offline models just like that. Yes, I think that would be very important.
Could I add to this question? I think that, Phil, I think your question is super important and one that I'm thinking a lot about right now. And so with the carbon export, that's exactly what we're trying to do, that if we can demonstrate in a simpler model that variable stoichiometry is really central to, carbon export patterns, which is what sort of one of the key properties that people think about in CMIP type models. Maybe we can convince modelers that in the CMIP community that they should think about variable stoichiometry. But of course, they're not going to take a 14-parameter trait-based model into their CMIP model, right? Then we would sort of occupy half their parameter space in the biological oceanography module. And so what we've then derived is sort of what we call a meta model, that is a very simple statistical model just based on temperature and nutrients. And that we have sort of like some traction, like then now we're down to 2 parameters that we can convince like an ocean biogeochemical model to incorporate by having demonstrated that has a real impact on carbon export or something like important for large scale. And I feel like maybe, so that's sort of what we're trying to brainstorm with the CMIP model people. What kind of data should we demonstrate, and what kind of data should we produce before you're convinced that you should spend 2 years on optimizing your large biogeochemical model for bringing in this property, and that's sort of like the process.
Okay, I guess this is for Adam or whoever else wants to answer. So it's a question of life and death. Can you comment on the role of detritus in driving variable stoichiometry across these large latitudinal gradients?
Yeah, so that's a really complex question. To what extent is sort of like particle export driven by detrital matter, and does knowing that a part of it is detrital, is that important? It only becomes important if there are sort of separate processes that drives the elemental composition, for example, of detrital matter before it gets exported. So if it's just a phytoplankton that was first alive and then is dead and then it gets exported without changing its composition, right? We could say, well, that doesn't really matter. But if something happens to that material as it sort of sits around at bats for 3 months before it drops down, then it does matter. And also, is it like 5% that's detrital, or is it 90% that's detrital?